Okay, well, I think I will begin just in the interest of keeping us on track. So today is kind of like the focus of today is evolutionary therapy, and well, there's multiple reasons why we wanted to have a focus on evolutionary therapy, but one of the big ones is that a large part of the funding for this meeting is coming from the Jacobson Foundation, who gave a substantial donation to our Centre of Excellence for Evolutionary Therapy. And because of the profits from it, we're able to pay for most of the festivities that we've had over the last couple of days and will continue to have. So big shout out to the Jacobson Foundation and thanks for believing in evolutionary therapy. So obviously, evolutionary therapy is something that there's an interest that's growing across the world. It's something that has origins outside of Moffitt, but in many ways, the first successful trial, is something that was kind of driven by Bob Gantamy and Jinsong Zhang, and you're going to hear from both of those. And of course, Joel was heavily involved too. But alongside this sort of clinical, practical reality, there's been a kind of community of theoreticians working with experimental colleagues and driving clinical trials, trying to understand how to, do better evolutionary therapies and understand the origins of that, come up with better strategies, really just using simple mathematics. And I think the work of Rob Noble really speaks to that beautifully. And I know he's going to talk about more than that, but I think that paper of his with Yannick is something very special and influenced our own work in many ways. So Rob, I guess, has a bit of an unusual background because his PhD wasn't in mathematical biology, it was infectious disease at Oxford. But he got interested in sort of mathematical oncology, and in part, believe it or not, because he attended one of the IMO workshops, and that was in
2014?
2014. And that was at the time we had the Ebor houses. So he had the Ebor house experience. And for those of you that know, you know. And if you don't, well, it's unfortunate. But the Ebor houses were a group of houses that we put all of the participants in, and they were connected through the back doors. And so you could, the teams almost sort of spontaneously chose a house and sort of would work there till the late hours and talk and do science and drink and have fun. And so I think, that maybe had some impact. But of course, mathematically, evolutionary therapies are just interesting and there's different ways to tackle them. And so without delaying any further, I'm really looking forward to hearing what Rob has to say. So let's give him a big warm welcome.
Thanks very much, Sandy. It's wonderful to be here. So thank you very much for inviting me. This is my favorite community, so it's fantastic to see you all here. So I'm going to talk about the title here says supporting clinical trials. Now, at the moment, I'm not actively involved in clinical trials, but there are many going on, as you can see in this list here. This is something I compiled recently. I can't guarantee it's 100% correct, but at least it gives you an idea of what's going on. Most of these trials are organized by Moffitt. There are a couple of exceptions, and you can see those are particularly the largest trials in Australia and the Netherlands and in London. So this is very encouraging for the field, I think, that it is not just one center, but other people are also catching on to this idea. And we're expecting a lot of data to come through in the next few years. But if you look at this array of different therapies and different cancer types, it's difficult to find a clear pattern in where these therapies are being targeted. So this is where mathematical modeling can support to say perhaps this approach is most suitable for a particular cancer type or a particular treatment, and to design the protocols that we think are going to be most effective and to help with other aspects of trial design. So that's the inspiration for the work that I'm going to present. So the goals for this talk then, the first thing I'm focusing, and I know Sandy wanted me to do this as well, on new unpublished results. A lot of this I've never presented before, so this is a completely new talk. Running theme will be considering what features we should include in these models. So there have been a lot of models here looking at the microenvironment, ecological models. I will say from the start, that's not what I'm going to present. So this is a big caveat that I'm more talking about evolutionary dynamics. These are the simple evolutionary dynamics models that we might use to predict outcomes of these interventions. And then I'm going to start thinking about, given what we might include in these models, and I'm going to be giving examples of that, how we might design models that we can apply across many cancer types, across many treatment types, so that we can rationalize where we choose to conduct trials for each therapy. Or you could say within this cohort of patients, these are the ones for which this strategy is most appropriate. These ones, we try this other strategy. So what do we include in a mathematical model of evolutionary dynamics during treatment? Well, the first thing we're going to have to include is some notion of growth. We're going to have our population of tumor cells either going up or going down over time in the absence or in the presence of treatment, right? That's the most basic aspect. What else could we include? Well, we're going to have a little game. So this is something you can play along. If you see other models today, you can play this same little game. Here are some of the things we can include. So subclonal interactions, large population, nice to have if you're modeling large tumors, three-dimensional microenvironmental components, which is obviously a large one, spatial structure, stochastic growth. Is it a tractable mathematical model? And do we have stochastic mutations? So feel free to take your own copy and play along as you see other models. I will be filling, well, Bingo here will be helping me fill it in along the way. So you can critique if you think I'm getting it right. All right, so I'm going to talk about three strategies. The first one is bipolar androgen therapy. This is a strategy for treating prostate cancer. And as you will have seen, many of the trials that are ongoing are in prostate cancer and rely on manipulating sensitivity to androgens, so testosterone. So a conventional treatment for prostate cancer is androgen deprivation therapy. The idea is that you starve the cancer cells of the testosterone they need to proliferate. These cancer cells have become dependent on testosterone, so you remove that. The problem is that cells adapt and some of them become castrate resistant, which means that they can grow even under very low levels of testosterone. They become extremely sensitive to testosterone. But it turns out that they are so sensitive that if you give them very high levels of testosterone, that's bad for them because they over-respond. They're overly sensitive to it. And this suggests a treatment strategy. So the idea of bipolar androgen therapy is that you alternate between these near castrate levels, extremely low, and very high testosterone levels, a few times in excess of normal physiological levels. And by doing that, you can target the two phenotypes by yo-yoing between very low and very high. And this has been proposed as a way to not only potentially prolong survival, but also to improve quality of life because androgen deprivation therapy has quite severe side effects and potentially also restore sensitivity to ADT so that you could do BAT for a while and then you could switch back to ADT. There have been various small clinical trials. They're listed along the bottom there. So I think about half a dozen small trials, most recent reported in 2021. In those trials, what they did is they used monthly injections. So there were big spikes in testosterone. Then the level went down to near castrate levels, and then they had another bolus the next month. So how are we going to model it? This is a very simple mathematical model. This bipolar androgen therapy has not been modeled before. So what we're doing here is just a first take to get an idea of what we can do. All we have here are a couple of ordinary differential equations, and we've got growth rates. The growth rates depend on the androgen, testosterone, which is A. Okay, so we've got growth rates GH and GR. Both of them are dependent on testosterone. And assume that these growth rates, the dependence is such that that would be an effective strategy. So this is what those growth rates would look like. They have two components, obviously. So you have the cell proliferation rate on the left, then you have the death rate. And for the hormone naive cells, which are in blue, their growth rate increases with the testosterone level and their death rate goes down. So these ones, ADT is a good strategy, just reduce testosterone. For the castrate resistant, up to a certain level, they like the testosterone, but once it goes above normal physiological levels approximately, then they start to suffer. Their proliferation rate goes down, their death rate goes up. So this is consistent with the BAT strategy that we could yo-yo between extremes, very low, very high, and we'd alternately target the two types. This work is led by Blair, by the way, who just finished a PhD in my group. So this is all his stuff that I'm presenting. How can we summarize that? Well, from these death and proliferation rates, we can get the net growth rate of each type, and then we can plot those two growth rates against each other. So the black curve here, you travel the black curve as you increase A, the testosterone level. So if you keep the testosterone level constant, you can only traverse this black curve. But if you vary A over time, so I'll try and pointer here. So if you vary A over time, for example, if you were to have a level of A which corresponded to this point here, and another which corresponded to this point here, and you yo-yo between those two, then on average, you'd occupy a position in the middle. So by choosing points on the black curve, you can occupy anything inside this space, on average. And that means you can do better than the black curve by alternating. Okay? And best of all would be to alternate between the extremes here, and then you'd be somewhere on this orange line here. And in particular, you can see that if you are somewhere around here, then your average growth rate for both GH and GR is close to 0, and so that you assume the optimal solution lies roughly around here. Okay? But you don't have to be approximate about it. You can calculate this exactly. So this is what Blair did. So as a first step, let's just assume that we can switch instantaneously between the minimal and maximal points. So we switch between this extreme and this extreme. And the testosterone just goes instantaneously up or down at our command. We make some other assumptions. These are mathematical assumptions, just to ensure that that really is the optimal strategy, and that the optimal strategy is not simply to stay at one extreme or the other. So these are assumptions which hold for this system that we're not going to go into. So what do you get? Well, you get this nice analytical solution. And it turns out, which we didn't expect naively at the beginning, that this optimal proportion of time to spend at the minimal testosterone level doesn't depend only on the growth rates. It also depends on your initial population sizes, H0 and R0. It depends on the slope of that orange line, and it depends on the total time that you treat for. Well, that's very nice, but it's a very basic assumption that you can switch instantaneously. In reality, if you're switching from the low level to the high level, you can do that pretty much instantaneously because you give an injection. So the testosterone level goes up. But it takes a while for the testosterone then to decline. So in reality, what happens is you jump from this point to this point, but then moving back, you have a black line. And that's not ideal, because the longer you spend on this black line, the more you move away from the orange curve on average, so that you're occupying an internal point, which is suboptimal. So how do we allow for that? Well, you can do simulations, but as a first step, what you can do is use a step function. You just assume that every time you switch to a high level, before you go back to the low level, you have to spend some amount of time at an intermediate suboptimal level. So we're approximating an exponential decay curve with a step function. And the reason we do that is because then you can get analytical solutions out and you get this extra term here, which is allowing for the time you spend at this intermediate testosterone level. And that makes things a bit more interesting because then we can think about how often do we want to switch. If you imagine in the instantaneous switching model, it doesn't matter how often you switch. Your final outcome will be the same because your average growth rate will be the same. In this case, you pay a penalty every time you switch. You pay a penalty because you have to stop off at the intermediate level on the way back. So that would tell you we don't want to switch very often. On the other hand, there's a penalty for infrequent switching, which is that your population size is going to yo-yo massively. So on average, your tumor size will be bigger because the oscillations are bigger. So that gives us a bit of a trade-off. If we're looking at how often we should switch between very high and very low levels, what we see is given the number of switches, the optimal for the final cell number is to switch less often because you're paying less penalty for being at the intermediate levels. But if we consider the average tumor size, and mean cell number, then there is a trade-off here between big oscillations and big final size, and you see that infrequent switching is optimal. So this is the kind of strategy we end up with. So what Blair did was he used his formula there for the step function, and then he ran simulations based on those parameter values with exponential decay to see whether the exponential decay made much difference. And what you see here is it's a good fit. So you can use the step function to get predictions that work for the exponential decay model. So what you get is this yo-yoing of the two types, and overall your population size is going up, at least for these parameter values, but it's going up more slowly than it would under other strategies. A difference between this strategy, this optimal strategy, and what's been tried in the trials is that in the trials they had monthly injections, and here what we're proposing is that if you wanted to optimize this treatment, you would maintain the high testosterone for some longer period and then remove it. So we're not saying that this is something you could necessarily exactly enact in a trial because there are also side effects for having very high testosterone levels. There are risks associated with that, but at least it gives you an idea of the scope for improvement over the BAT protocol in previous trials, which was not based on any modeling whatsoever. So what you find is that if we compare for this parameter set, ADT, we get this pale blue curve here. The standard BAT protocol is in green here. So the standard BAT protocol is, in terms of final tumor size, it's similar to ADT. But as I say, in terms of side effects and in terms of potential resensitization to ADT, it's better. And therefore, at least it's no worse. But we find that there is scope to do better by about an order of magnitude than the previous BAT protocol. And the next step would be to look at the constraints imposed by the protocols that are actually clinically feasible. Okay, so that's BAT. Let's look at another strategy that has been proposed, again, by someone in this room. Multi-strike therapy, also known as extinction therapy. We're calling it multi-strike therapy here just to be precise about how we're doing this. So this is what Bob Gatenby and colleagues proposed a few years ago. The idea is to exploit the vulnerability of small populations to extinction. So essentially what we're doing is we're applying one therapy, which reduces the population size of the tumor. And then rather than waiting to see whether that tumor is going to relapse through resistance to the first therapy, we apply a second treatment, even while the first treatment, the tumor is undetectable in response to the first treatment. And the idea is that you exploit this sensitivity of small populations to stochastic extinction. And potentially in this way, you can integrate therapies that by themselves wouldn't be very effective. So maybe you can reduce doses, you can use treatments that have fewer side effects and so on. So this is the big question. We have a population that is declining due to your first treatment here. If it relapses, it's going to climb back like this. And there is some nadir here, which we call N min, the minimal population size. And the question is, when should you switch from this first treatment to a second treatment? So we're not comparing here with combination therapy. We're not. I could go into that if you like in the questions, but we're just considering that your protocol is to treat with one treatment, then switch at some point. And the question is, when do you switch? Should you switch sometime around the nadir? Should you switch a bit before, a bit after? That's our question. How do we model that? Well, I was very lucky to be working with two talented students here, Srishti and Arman, who went to town on this mathematically. And what we used is a deterministic model. So you assume that you have some initial population that comprises sensitive cells, resistant to the first treatment, resistant to the second, no doubly resistant cells. Because if you had a lot of doubly resistant cells initially, your strategy is doomed to failure anyway. This wouldn't be a good strategy to try. Then you apply the first treatment. This is environment 1 during the first treatment. Your population size goes down. Then if you didn't apply any second treatment, this is what would happen. If you had R1 cells surviving, you would get relapse. Or potentially you can get population extinction. And the question is, when should we apply this second treatment? So to address this, we used a deterministic model to model this initial decay, an ODE model, and then a stochastic model to look at what happens when the population size is small. So the framework here is evolutionary rescue theory. Here we have the ODEs. Here we have the populations and all these mutation rates. So here we've got growth terms, mutational terms. We have density dependence here. It turns out density dependence doesn't matter much, but we put it in anyway. Within the second treatment phase, where we're looking at the stochastic probability of extinction, then we have, dependent on the treatment switch time, tau, we have a probability of extinction due to the standing genetic variation, the resistance that was there when you started, and the probability of extinction due to the de novo mutations. You multiply those together, you get the overall probability of extinction. So to find those probabilities, you use evolutionary rescue theory. There's a big literature on evolutionary rescue theory applied in all kinds of domains, sometimes trying to preserve populations, sometimes trying to get rid of them. So we have all the mathematical theory that we could adapt. So what do we have? In summary, we've got the analytical solution of the approximate model. So we could get really nice analytical solutions out here. Extinction probabilities using the evolutionary rescue theory. We also used agent-based modeling. So we had stochastic simulations to check that our results were valid and accurate, and a lot of sensitivity analysis and all that. So what the bingo tells us is that we've got a bunch of features here that we'd like to have. So compared to before, we've got subclonal interactions, we've got stochastic mutations, still tractable, and we've got stochastic growth. So what you find is that according to this framework, the optimal time to treat, so here we have the extinction probability versus the size of the tumor at which you switch. This is the nadir, N min, and you find that to optimize extinction probability, you treat very close to the nadir. But if you're unsure exactly where that nadir is, which would be the case in the clinic usually because the tumor is undetectable at this point, then it is better to switch a bit after than a bit before. Okay, the blue line is above the black line. Oh, by the way, the points here, those are stochastic simulations with the Gillespie algorithm, and the curves are analytical solutions. So you can see the analytical solutions do hold. So this is, the result that is better to treat a bit too early than, a bit too late than too early. The reason for that is the standing genetic variation. If you didn't have any R2 cells, so if you had no standing resistance to the second therapy initially, then there's not much difference whether you treat before or after. So this is all to do with the fact that when you're treating initially, your R2 population is sensitive to the first treatment. So that population is going down, and it turns out that that means that it's actually better to treat later than earlier. There are a lot of other results in this paper. So the paper has just been accepted a couple of days ago. It will be coming out very soon. There is an earlier version of bioRxiv which has most of the same results. Some of the other results, we find that you can get away with lower efficacy treatments, which is very encouraging. The cost of resistance is not so important. Carrying capacity doesn't matter too much. We put density dependence in, but we can show that it doesn't matter too much. The extinction probability increases with the death rate and the turnover. The headline is that this approach is, however, only feasible in small tumors. And we're talking very small. But that's two-strike therapy. So as we explain in the paper, the potential would be to apply multiple strikes, and that will be the next step to model what happens if you either yo-yo between two treatments or you apply multiple treatments in sequence, and potentially you could then treat much larger tumors. Also, in this paper, we are very conservative in our assumptions. So in particular, we start off with very high levels of resistance, which are larger than you would get in mutation selection balance. So we are trying to be very conservative with more perhaps realistic parameter values. You might find that this is a bit more effective. Okay, strategy 3 is one that has already been spoken about, which is tumor containment or adaptive therapy. So this has been introduced very nicely by previous speakers. The idea here is, as proposed by Bob Gatenby and colleagues, and around the same time by a couple of mathematicians from Oxford, that we can contain tumors. The extinction therapy, multi-strike therapy case that I just presented as strategy 2, this is what you'd like to do if you know from past experience or you have other good reason to suppose that there's a good chance you can eliminate the tumor. If you can go for a cure, go for the extinction therapy approach. But if you have reason to believe that a cure is very unlikely, then a better strategy would be to contain the tumor and to try and prolong survival as long as you can, rather than failing to cure. So that's the idea of tumor containment or adaptive therapy. So the basic idea is to use lower doses, doses that are insufficient to shrink the tumor, but are just enough to maintain it at a constant population size. And by doing this, you exploit competition between sensitive and resistant cells, and you use the sensitive cells to contain the resistant cells. Because if we've got highly resistant cells, there is nothing else we can do to stop them growing apart from exploiting that competition. That's the idea. So tumor containment is this basic idea that you freeze the tumor, and adaptive therapy is a practical strategy for doing this, which is that initially you won't know what that dose is to contain. So what you do is you try something and then you modulate either the dose or the gaps between doses in order to find that level through trial and improvement and maintain the tumor at a certain level. So that's the basic idea. And so Bob Gatenby and colleagues here at Moffitt have run with this idea and done a huge amount of work on it. My collaborator Yannick Biersat and I, as Sandy mentioned, we published a paper a few years ago where we looked at this in a very general context. We said if you've got a general model where you have these two cell types, sensitive and highly resistant, they have growth rates, GS and GR. These growth rates are dependent on the number of sensitive and resistant cells. So they're either frequency or density dependent. And the sensitive cell growth rate also depends on the drug dose, of course. If you make a couple of simple assumptions, so GS is non-increasing in C. In other words, the more treatment you give, the more you hit the sensitive cells, which is usually true. And the growth rate of resistant cells is non-increasing in S. In other words, the more sensitive cells you have, the slower the resistant cells grow. So if you make just these very basic assumptions, you don't have to put any particular form in here. So you don't have to assume that you've got Compertzian growth, logistic growth, whatever. It doesn't matter. You can use an agent-based model as long as these assumptions hold, you can show that generally the optimal strategy is to maintain the tumor burden as high as possible while sensitive cells remain, and we call this ideal containment. So the best you can do is to find the treatment level that keeps the tumor exactly flat at the highest burden that can be tolerated. So this is obviously a clinical decision. You say, what is that burden that's acceptable? That's a clinical decision rather than a theoretical one. But if there is such a burden, then you want to maintain as close to that burden as possible. And the worst option is to maximize the cure rate under these assumptions. So that Yanni really did the heavy lifting there on the mathematical side. You can show that holds very generally. It doesn't really matter what model you use in terms of this qualitative result. But it does matter quantitatively. So on the left here are growth curves for populations following three different growth laws: von Bertalanffy, Gompertzi, and logistic. These are three commonly used growth laws to model tumor growth. And I've picked the parameter values here such that within this range of tumor sizes, these growth curves look pretty similar. So you can imagine that if you were recording the size of these tumors, you wouldn't be able to infer which growth law is going on, right? These tumors seem to be growing similarly. So you might arbitrarily pick any of these growth laws and say, oh, it's a pretty good fit to logistics, so we'll use a logistic growth curve. The problem is that your prediction of the relative benefit of adaptive therapy compared to MTD massively depends on which of these growth laws you decide to use. So logistic is the most pessimistic, von Bertalanffy is very optimistic, and Gompertzi is somewhere in the middle. So it is a real headline from this paper is that the clinical gains strongly depend on the intensity of the competition. This is the most important factor, right? How do the growth terms, if we go back to the equations here, what is this dependence? How does GS depend on SNR? How does GR depend on SNR? And the problem is we don't really know. So we can try different things and we could say, well, if it's like this, then you get this. And the reason for this is that in reality, this competition depends also on your spatial context. So suppose these are two tumors, one's bigger than the other, and you have some resistant cell populations A, B, and C. If you were to apply this tumor containment strategy, which of these is going to actually turn out best, do you expect? Well, I think B is going to be pretty poor because this population size, this B population is very close to escaping the tumor. And once it starts to outgrow here, you no longer have the competition. The treatment's going to fail. This is better, but I think that C is probably better than B, even though this tumor is smaller. Whereas our non-spatial model would tell us that this is generally better than this. It doesn't take into account this spatial context. So how can we allow for that spatial context? How can we capture that within a model? Well, we can't do it within our non-spatial model. At least I mean, we could do it on average perhaps, but we'd like to do something a bit better than that. Well, of course, you go to an agent-based model, of course, right? We've seen a lot of agent-based models here. So at this point, I want to say, yeah, agent-based models, they've got a lot going for them. We can have the interactions, we can have the microenvironment, stochastic growth, stochastic mutation, spatial structure. We can put all of that into it. But there are limitations too. In an agent-based model, you generally can't go to very large populations. You can't model a whole large tumor at the size that is usually detectable. Three-dimensional is tricky. You can do three-dimensional, but then again, you compromise on the radius of the tumor you can simulate, and they're generally not tractable, which means that we can simulate something, but then we can't really predict what would happen if we use different parameter values, if we implemented slightly different rules for all these different components and so on. But before I go on here, I want to say I still do like agent-based modeling, and I've done a bunch of it. So it's not that I'm knocking agent-based modeling. I'm just going to present an alternative. And the last paper here is even a how-to guide we did for agent-based modeling of tumor evolution. So yes, it's not that I think that this is a bad approach. It's just that I think we can think of alternatives as well. So one of the drawbacks of agent-based modeling is that because it's stochastic and because all of these properties are emergent, you get huge variation in outcomes. So that's a feature because it represents biological diversity, but it also makes it hard to interpret the results because this is an example where I set up simulations, spatial simulations here in 2D, and these two replicates had exactly the same parameter values, same initial condition, but they follow very different evolutionary trajectories. Here you get a couple of selective sweeps. So here we're looking at time on the x-axis, frequency on the y-axis. We've already seen a few of these Miller plots. And the populations grow out of their parents, right? So it starts off as this grayish brown type, then a blue clone emerges and sweeps through because it's fitter, as you can see at the bottom here. Then a green type emerges out of the blue and takes over. Towards the end, there's a pink type that's busy taking over as well. So we get these sequential selective sweeps. Here, it just happened that no very fit mutants arose early on. So instead, what we got is what we call clonal interference, where multiple mutations coexist with similar fitness values, and they just run up against each other, and you don't get sweeps. So I did a lot of simulations for this paper a few years ago, but they were just simulations. I mean, there's a huge number of simulations, but they are still just simulations. And what I wanted to understand is how often could we expect to get this selective sweep scenario versus clone interference? Could you do something more than just run simulations? So this is how we went about doing that. So what we're doing here is we're starting with the idea of a microscopic model. This is what you would have in your agent-based model. So in this case, a spatial Moran process. Dominic introduced some of these ideas very nicely in his talk previously. So here we have demes, as Dominic explained, where we have well-mixed populations within a patch in space. Those populations compete. So it's generally a small population drift as well as selection going on. And then you have dispersal between the demes, between the patches. And we assume that it's nearest neighbor dispersal. Okay, so that's what you could simulate, and that's what we do simulate. So I've got software that I developed that simulates this process very efficiently, and that's what I used to get the previous results. But we also know, we've seen examples of PDEs here, that from this process, you can obtain PDE approximations by going from discrete to continuous space. In particular, you can get Fisher waves or the FKPP equation, which tells you there will be a traveling wave. So one type, the fitter type, will spread through the less fit type at a certain speed, and you can derive that speed from the parameters of your microscopic model, right? You can get your diffusion coefficient. Now, that's fine. So you could run PDEs, but if you've got a traveling wave solution and you don't care too much about the wavefront, so you don't care too much about what's going on where these two types are interacting, you just say one type is spreading into the other at a certain speed, you could just model that. So what we go from then is to this macroscopic model. Here we're assuming continuous space and constant expansion speed. So we have a ball of cells that is expanding in space, and then we have mutations arising and they spread within the initial population at a certain speed. Now, the advantage of that is that this becomes tractable. So I'll show you how. So on our bingo card here, now we have subclonal interactions, large population. We can have arbitrarily large because this is all continuous. We can make it three-dimensional. That's trivial. We have spatial structure. We have stochastic mutations, and it's tractable. We lose stochastic growth because we're assuming that the growth, if these populate, we can allow for the probability that they establish rather than go stochastic extinct. But after that, we assume they grow deterministically. That's the compromise here. So we have this simple model where we just have a ball of cells, a continuous ball growing in space in 1, 2, or 3 dimensions. And it's growing in this simple model at a constant radial speed. A mutant arises at a random time and location, this inhomogeneous Poisson process, and then that grows at radial speed cm within the wild type. So we're leaving out all of the interesting ecology, which is the topic of so many talks here. We're putting all that aside. The reason for that is because we're interested in this question of when do you get selective sweeps? And so this setup actually gives us the best case scenario for selective sweeps. We're setting up a model that actually allows selective sweeps to happen. If we set up different ecology, if we said there's this region here where the mutant is actually really unfit, then trivially you wouldn't get selective sweeps. So if you assume that there are different environmental niches here, or there are physical barriers, then you would not get selective sweeps for biological reasons, and it's not worth modeling. So what we're trying to say here is, can we set up a model that is going to give us an upper bound on the sweep probability? So this is what we do. We have this model, and we're looking at whether you get a selective sweep or clonal interference. Those are the two possible outcomes. In terms of the phylogenetic tree, this says that is the first event in your phylogenetic tree from the root to the first populations, is that linear or is it branching? Okay, and this is the Muller plot. So I hope Heiko is okay with us having three parameters. We have two speeds here: the speed of the mutant, the speed of the wild type, and we have a mutation rate. That's it. Okay, so it's a very simple model. And we have the spatial dimension, 1, 2, 3. So what do we do? Well, you take a stochastic processes approach, which is there. Okay, so yeah, you find the probability distribution, the probability density functions, and then you do some integration, and there's a lot of maths in this paper. But the final outcome is actually really neat, because here we have the ratio of fitness difference between the mutant and the wild type. You could also plot this in terms of speed, but here we've assumed the spatial Moran process, so we can parameterize this in terms of fitness difference. You have the probability of a sweep. So the solid curve here is the exact solution. The dashed line here is an approximation, which turns out to be pretty good and is an upper bound. And this approximation is beautifully simple. I hope you agree, which is that the probability of a selective sweep in d dimensions is approximately 1 minus the ratio of the speeds to the power of d. Couldn't be much simpler, right? And that turns out to be a nice upper bound that's close to the exact solution. The points here are from agent-based simulations. So you find that, yes, it does hold, even though our agent-based models have discrete space and they have various other properties that don't hold for our analytical model. The other thing you might notice is that originally we had 3 parameters. We've only got 2 left. Where's the mutation rate? Well, it turns out that this sweep probability is independent of the mutation rate. And you can see here we varied the mutation rate over 2 orders of magnitude to prove that. And yeah, so this neat solution here holds independent of your mutation rate. This is 2-dimensional, by the way. We also get results for 1D and 3D, and Alex also looked at all kinds of alternative growth models. So exponential, logistic, boundary growth, you name it. But we find that this is a useful general result that actually sweeps are pretty rare. So why am I talking about this? Because this actually is not related directly to therapy. Well, the idea is that we could take that model, which we've already analyzed, and we could do something similar for tumor containment. And that's what my PhD student Kate has begun doing. So these are really preliminary results from Kate. So we have the same setup here, that we've got a wild type tumor that's expanding at a constant radial speed. Resistant mutations, in this case, multiple mutations arise at various points and times. Then we apply treatment. Treatment halts the growth. So these arrows disappear, right? The growth of the wild type has stopped, but the resistant populations keep growing. If they didn't, we'd be able to contain indefinitely. So we assume that the resistant mutants keep expanding at some slow rate. And then eventually, when one reaches the boundary, at that point, we can no longer contain it. We say that's the end point. So we're interested in how long we can delay this endpoint. So what Kate's begun doing is analyzing this model. And the simplest case, she's taken a fairly reasonable resistant mutation rate. She's calibrated the growth rates for a reasonable growth time to 1 centimeter radius. Again, assuming here that growth is polynomial, which I know is not wholly realistic. And you can use analytical prediction and simulations, and you find the agreement's very good. The main thing is, unsurprisingly, that you get massively more mutants in 3D than 2D. But perhaps more interestingly, if you look at the distribution of these mutations, you find that in 3D, the mutants tend to be closer to the boundary than in 2D. So this is a bit of a caveat for the use of 2D simulations to model 3D systems, which is something that I've done a lot, but there are caveats here that you know there are properties of 3D that are different from 2D. So this all looks very pessimistic for tumor containment or adaptive therapy, right? And indeed, if you try to find the predicted benefits, the time until the resistant cell population escapes, then you find that in 3D, it's much worse than in 1D or 2D. Under these assumptions, right? This is the first step. Kate also looked at the exponential growth model. It's not quite as bad there, but again, 3D is worse than 1D or 2D. And she can look at heat maps and so on. All right, so again, Kate just finished her first year. These are preliminary results. So there are a lot of caveats we can discuss later, but this is the direction we're going. So what are we doing here? We're developing a macroscopic spatial model. The idea is that then we can model arbitrarily large population sizes, and we can do it in a way that is tractable. We have very few parameters. We can parameterize these models in terms of only the expansion speeds and the mutation rates. Those are the only parameters we care about. How do we derive those expansion speeds? You can use clinical and experimental data. You can also use these microscopic mathematical models. So if you've got an ABM that has all of this complicated ecology, and you find that the macroscopic pattern is that population A expands into population B at a certain speed, then you use that to parameterize this model and say, we're just going to model that growth, right? So this is not doing away with microscopic models. It's just taking their results and then saying, okay, effectively what's happening at the large scale. So we get a broad view of what's going on. All right, so everything I just presented is very speculative, right? There are no parameter values there. There are lots of caveats about what assumptions we're making. So at this point, you should say, oh, where's the data, right? How are you going to actually parameterize this? Well, the good thing is I have collaborators. So I'm currently on sabbatical in Montpellier, and in Montpellier, I have collaborators I've been working with for 11 years, more than 11 years. And previously, we published some results on adaptive therapy and tumor spheroids. There's now a new generation of PhD students, including Alex here, who's been doing some fantastic experiments that I'm going to present now. And what we're now looking at is a lung adenocarcinoma cell line with these EGFR inhibitors, alotinib and osimertinib. So this is very much related to what Maxi presented on the first day. So it's great that he introduced a lot of the ideas there. It's a similar kind of setup. So Alex has 3 cell lines. He has the PC9, which is sensitive to both of these inhibitors. He has a cell line that's resistant only to alotinib. You can see there's a massive difference here in the IC50. And he has doubly resistant cells as well, which have strong resistance to both treatments. Now, previously, when we were doing these adaptive therapy experiments that we published in 2017, I think, we were looking at a investigative CDK2 inhibitor. Here, we're looking at these targeted therapies that are clinically applicable. So that's why we're especially excited about these results. So the first thing you can do is you can do monolayer growth cultures to see how fast do these 3 cell lines grow and what are their carrying capacities. So there are different y-axes here. What you see is that the headline is that the carrying capacity for the resistant cells is the doubly resistant is substantially lower. Initial exponential growth rate is lower as well. So they are paying a cost for being doubly resistant. So from that, you could predict that they would lose out if they were in competition, but that's not necessarily going to hold given the way cells interact when you put them together. So you have to do the competition assays. So this is what Alex did. He did a huge number of assays in 2D culture. He did cell sorting at the end because these cell lines are fluorescently labeled red and green. So you get these different subpopulations. From that, you get the frequencies of sensitive and resistant. From that, you do a bit of maths. You do the log of the frequency ratio, which is a logit transform. for each of the time points, and here for each of the treatment conditions, different doses. And then from the gradient of these lines, you get a selection coefficient. So the selection coefficient is the difference in fitness. So what do you find? You find that depending on your initial frequency of resistance, the cost of resistance varies. So there's frequency dependence here. So the dashed line is what you would predict simply from the monoculture exponential growth rates. But when you put these two cell lines together, you see that the resistant cells actually prefer being surrounded by sensitive cells. And that could make sense because, as we've seen on some of the posters as well, there can be interactions between these two cell types that the resistant cells perhaps are producing some factor that harms both them and the sensitive cells. The sensitive cells are nicer. Or it could be in how they use up resources. We haven't got that far yet. So Alex repeated this experiment in low density, because in the high density case, you have the effects of frequency dependence and density dependence. He then looked at what happens if you only have frequency dependence. Same thing. All right, what about 3D? Well, the nice thing about 3D, these spheroids, is that at the IGMM, they have this great confocal microscope. So this allows them to take these images of the tumor spheroids, and each of these little dots here is a cell. So what they do is they take images all the way through the spheroid, and then they use software to exactly identify each individual cell, and this allows you to exactly quantify how many resistant and sensitive cells you have, and also their spatial context. So this is what you get. So this is a competition in 3D, in spheroids, with the same cell lines, where we have initially 10% resistant cells. And here we've separated the channels, so these are the sensitive cells, these are the resistant. The encouraging thing is that the resistant cells here are mostly quite far from the boundary. But this is a different setup to Kate's model. So in Kate's model, resistance emerges through mutation as the tumor grows. In these spheroids, you initially mix the cell lines together, so you have slightly different initial conditions. But with that caveat, that's what we're looking at here. And with this software, you can separate these different populations here and identify exactly where they are and how large they are and so on. If you start with a 50-50 mix, again, the resistant cells gets left behind to some point, to some extent. They're not right up on the boundary, are less fit. They're getting left behind as the spheroid grows. So this is actually a bit more encouraging for adaptive therapy. What's the frequency dependence? Well, we can look at that exactly as we did in the 2D. And the interesting thing is you find the opposite result. So this is what we saw in 2D. In 2D, as resistant cells become more frequent, their fitness goes down. So they like to be surrounded by sensitive cells. In 3D, it's the opposite. They like to be surrounded by their own type. That makes sense. If they are less fit than the sensitive cells, then they fall behind as the 3D population is growing. Whereas if they're surrounded by their own type, then they're okay. So that, again, this is a caveat to taking 2D results and putting them into 3D. So these are the brand new slides that I just got yesterday. From results that Alex did very recently, which is using osimertinib at a low dose on these spheroids to see if he can contain them. And indeed he can. So this is untreated on a linear scale at the top. What you see is that, so this is the average of 3 replicates. In the sensitive cells, in the absence of treatment, grow very nicely. The treatment is added at the dashed lines here. So in this case, it's just a control, but here is adding enough osimertinib that the sensitive population is kept more or less constant. And what you see is that the resistant cell population keeps low as well. The reason I included the log scale is you can see that the resistant cells are still growing. Okay, so you're not going to be able to contain forever like this. They are growing slowly through the sensitive cells, and that's as our model assumes. Alex has done the MTD experiment. but it's so fresh, he hasn't analyzed the results yet. So we don't have that explicit comparison here, but you can get an idea of what would happen in MTD by looking at what happened when he started with 90% resistant cells. They grew very nicely, thank you very much. So if you were to treat with MTD here, you can imagine that the resistant cells would increase. So you're getting a much better outcome with AT, adaptive therapy. All right, so what about mice? We've gone from 2D to 3D. What we really want for a preclinical trial is to do this in mice, and Alex is doing that. So he already has some results, and as I'm going to present here, again, very, very new results that we haven't presented before. So in this case, he had 10 mice for each of the cell lines. Without any treatment, if you just grow these tumors in mice, they do grow. They take very nicely. But again, these are different y-axes. On average, the resistant cell population grows much worse than the sensitive. So the fitness cost here in mice is bigger than it was in either of the culture conditions. But you also see a lot of variation here. Even though these are, nude mice, this is as controlled as you can get, you see a lot of variation in how these tumors grow. So in summary, fitness cost matters when you're predicting the quantitative outcomes of therapy, but you've got to be careful because this resistant cost depends on whether you're in 2D culture, spheroids, or in vivo, and the frequency dependence varies as well. We don't yet have enough data to find this correlation in vivo, but we would assume that it would be similar to the spheroids. All right, what if you treat? What if you treat these mouse tumors with osimertinib? Well, here are some initial results. So this is initial results for the sensitive cell line, and you find that the tumors grow, you apply osimertinib, you drive it down to a very low level. You can't quite cure it, but you can at least eliminate most of the cells. All right, so where are we going? This is what you want to see, right? This is the big slide I should be ending on. What happens if you do osimertinib adaptive therapy in mice with labeled resistant cells and you track the populations? Well, it's an ongoing experiment. So I don't have it quite yet, but the experiment is actually running right now. So we will have that soon. Okay, so I will just thank all the people who led this work. You've seen some of their pictures through the talk. And my collaborators in Montpellier on the bottom row on the right there. Yeah, I'll be happy to take questions. Thank you.
Okay, time for some questions. I'm sure there's many. Bob?
Thank you. Nice talk. Thank you for that. I wanted to point out something that although it's obvious, is not necessarily well known to empiricists, is that cultured cells are adapted to growing in the dish. No one really knows the selection forces that are in a dish, but they are very, very dependent on the media. Culture media typically contains 25 millimole glucose. Physiologic levels about 5. So you can manipulate the product, the components of the media to specifically select for things. And we found that if, for example, if you lower the glucose concentration, you get different results. Michelle Lockley, in a paper recently in Cancer Research, showed that the same thing is true if you lower or change the serum concentration. Mm-hmm. So I just wanted to point that out, that it's a factor that's not usually considered in these competition experiments.
Yeah, thank you. No, that is very important. I know that from bitter experience, from some previous experiments we had with another lab in the same institute, where the entire institute changed their supplier of medium. And suddenly the system that we had working beautifully stopped working, and it took months to get it working again. So yeah, that is an important caveat. And in the previous experiments, we did look at low oxygen, low serum, and so on. And I don't think they have results, but they might correct me and say that they do have some, but it's not here. But yeah, that might help explain the results, why the results are so different in 3D and in vivo.
Any other questions from the floor? I definitely have some, but I want to share.
Yeah, wonderful stuff. Thank you. I wanted to follow up. You mentioned 3D versus 2D, and there was a slide where you said that there were a lot more mutants when you grew it in 3D than in 2D. What is the mechanism of that?
It is simply that the control caters used there is simply the radius. So the population size is just massively larger in 3D than in 2D.
Oh, I see. It was
Yeah,
just
so you're just integrating over over a volume instead of an area. So it's,
right. Because I would have thought that if you compare equal population sizes, then 3D would probably have fewer mutants than 2D because it's a little bit more mixed.
That's true. So she could do that comparison, which I guess is relevant if you're thinking about agent-based models and how you go from saying we've simulated 1 million cells in 2D versus 3D. Yeah.
Right.
So I know we should do that. I'll suggest that too. Thank you.
Right.
Thank you for your talk. I have one quick question from your strategy number 2 when you have, I think you have 2 types of therapy on your tumor, and then you said the probability of extinction of the tumor is the multiplication of the probability of extinction for the first one and the second one. I was wondering to ask, how do you make sure that these 2 events are independent in order to calculate the probability of extinction of the tumor as the multiplication of these?
Right. So if I understand you, you're talking about the probability of extinction due to the first treatment alone. So for our parameter values, the probability of extinction if you only apply the first treatment is negligible. And we actually had some calculations for that because that's one of the things the reviewers asked for. It's very unlikely you're going to cure these tumors through just applying the first treatment. So the second treatment is critical. So you can actually neglect that probability because you are bound to have some resistance to the first treatment. Otherwise, you wouldn't even consider this, right? You'd say, okay, we've got a treatment that cures cancer. Let's just do that, right? And we only do this if that fails. But we're looking at parameter regimes where this 2-strike therapy is necessary, and in the regime where it might or might not work, which is the interesting case rather than the trivial cases at either end.
Okay, well, I'm going to ask a couple of questions. So first of all, thanks for pacing that nicely for my fragile brain this morning. I enjoyed that deeply. 2 questions. One, something that we're observing more and more is rather than a kind of mutational driven resistance, we're seeing a much more plastic type of resistance. And it isn't fixed and it's context dependent. And I know you didn't touch on that, but I just wondered what your thoughts were on how that might impact.
Yeah, I thought of putting a little asterisk at the beginning saying excluding plasticity because yeah, that is a massive caveat.
Yeah.
And that's also, for example, something that comes up in the case of extinction therapy is what if you have a plastic response instead? Now, it so happens that Bob and colleagues also have a preprint up, which is on a similar kind of model. In their case, they did stochastic simulations in SLIM, and they did look at what happens if you have a plastic response instead of rare mutations, and they found that the results are somewhat similar. So in that case, they had a trade-off. And so we didn't look at that explicitly, but we're very glad that they did, so that we can just cite their results and say, yeah, this has been looked at. But yes, generally, these are evolutionary dynamic models, and you have to make that decision. Do we have rare, strong mutations, or do we have plasticity and treat resistance and continuous trait?
Yeah. Or in fact, do we have both?
Yes.
Which is probably really what's there, right? Yeah. And then, so my second question was, love to see the fact that you're, explicitly including space and then trying to approximate it and coming up with some cool analytical results. But one of the kind of, I think, important things that sometimes we neglect when we're trying to model, let's say, in vitro systems. But when it comes to a patient with cancer, it's in an organ. And that organ has a normal tissue population that's pushing back against the tumor, isn't just in a void, right? And so that interaction with the normal homeostasis is going to change some of those competition dynamics. What are your thoughts on that?
So when we do the simulations for this agent, for this spatial Moran process, we start with a grid that is filled with normal cells. So we are actually explicitly modeling these. So I didn't go into the details there. If you don't do that, then what happens is you're growing into space. So your wild type tumor grows so fast that no resistant cells are ever going to catch up, right? Because no fitter cells are ever going to catch up because they're having to compete with the wild type tumor cells, whereas the wild type tumor cells are just growing into space. So they're zooming away. So in order to make this model interesting, you have to start with a grid that is filled with normal cells. You then start, these simulations start with a single fitter cancer cell, which has a slight selection advantage in the middle. Often those go stochastically extinct to restart with a different random seed. And then they have to grow by outcompeting the normal cells and invading the surrounding tissue. So then you have invasion of the tumor into normal tissue and invasion of mutants into the tumor.
I'm sorry. I just wanted to point out that the plasticity, which sounds like it was a hopeless, it added like a hopeless dynamic to this process, is actually vulnerable to epigenetic modifiers, and so you never get something for nothing. And so in this case, even though it's more plastic, it also has vulnerabilities to follow-on therapies that can exploit that evolutionary process. So I just wanted to add that it's not a hopeless thing, and it can be, I think, explicitly put into these models with a potential treatment agent. In fact, my lab is doing experiments on exactly that.
Oh, fantastic. Yeah, I look forward to seeing that.
Okay, let's thank Rob for a lovely presentation.